All right, next up, we're going to hear from Ryan Shewcraft from Bristol Myers Squibb Data Science Team and Megan Gross, Director at AiCure, on how to generate patient insights from speech-based biomarkers.
All right. So great talk, Daniel. I got some bad news for you. Psychiatric drug development is hard. In the decade leading up to 2016, only 6.2% of psychiatric drug candidates that started a phase 1 study were eventually approved by the FDA. And only oncology has a lower rate. So this really raises the question, why do so many clinical trials in psychiatric indications fail? And one reason I think for why that is that the brain is both biological and computational. So again, Daniel touched on this a little bit. Maybe I want to unpack it a little bit more here. So the brain's biological, we all know this. It's made of neurons, cells, neurotransmitters, all these sorts of biological bits. These bits get put together through, as Dan mentioned, synaptic plasticity. And in doing so, they build networks. And these networks instantiate algorithms which perform computations to transform our inputs, which are typically sensory, into outputs, our behaviors like talking, moving, breathing. These outputs are how we read out what our drugs, our molecules are doing to the biology. So these are the key things that we need to study to understand how the medicine is affecting the computation and thus affecting the outcomes. So to unpack this further, I really want to start with a personal story. On the bottom left is a picture of myself playing checkers against my grandfather, and this was maybe 4 or 6 months before he passed away from Alzheimer's. My grandfather was a great checkers player. He worked at the GM factory. At break time at lunch, he would play 10 people at once, beat all of them. When we played that day, he couldn't remember if he was playing black or white pieces. I kept having to say, Papa, no, you're black, move these pieces. Played 3 games, he beat me in every game. And this totally blew my mind as a first-year graduate student in neuroscience. So the first thing I did was went to my intro neuro textbook where I found this picture in the middle. This is the complex diagram of the memory circuit as we know it in the brain. And what you can see is there's a bunch of interconnected components. Each of these connected components and their sort of subnetworks sort of serve different forms of memory in the brain. And my grandfather, because of the way Alzheimer's pathology works, had lost almost all of his declarative memory. So his ability to remember names, know where he is, be able to say what he had for lunch yesterday. But the disease had spared a degree of his procedural memory, the circuit that affects his procedural memory. So he could still play checkers reasonably well. We gave him a little tiny keyboard. He was able to pick out a tune that he used to be able to play when he was younger. In psychiatric trials, as Daniel mentioned, structured clinical interviews are the endpoint that we measure. So I'm going to sort of skip through this, same sort of limitations as Daniel mentioned. So in addition to looking at ways to leverage large language models to sort of automatically score, give or score those clinical interviews, another area that we think that we could sort of leverage this digital biomarker technology is in spontaneous communication. So how are individuals communicating in their day-to-day lives? Obviously, there's a lot of information in sort of the language that is being used, the words that they're saying. So if they say, I'm really feeling bummed, the whole world's against me, you can probably assume that they're not having a great day or not in a great mental health state. But in addition, there's a lot of lower level information in this spontaneous communication that can tell us about how these circuits are impaired by the disease biology. So there are things like emotional valence. Individuals with anxiety might have higher levels of uncertainty or tentativeness that they're expressing in the way they communicate. There's also nonverbal communication, eye contact, facial expressivity. All of these things could be useful as sort of measurements of an individual's mental health state. Whoops, I hit the wrong button. So what we think is that there are a lot of digital biomarkers that could give us a way to extract information from spontaneous communication in sort of a quantitative way to allow us to better assess patients' mental health. And so with that, I'm going to hand it over to my colleague Megan, who can tell you how AiCure is doing just that.
All right. Hello. So my name is Megan Gross. I'm head of project delivery and research at AiCure. And AiCure, here we are, is a technology company that leverages computer vision and artificial intelligence to remotely capture and assess patient behavior, detect subtle cues of treatment response, and activate engagement between sites and participants. Our technology has 3 cores: a modular, highly configurable smartphone application that's custom built for your protocol and designed for your patients. We also have data intelligence and predictive analytics, which yield a holistic view of study data and clinical trial participant health and behavior through our visualization platform. And lastly, we also have digital biomarkers, which correlate behavior variables to disease state and treatment response. We started about 15 years ago with funding from the NIH, and we've since grown to deploy AiCure in more than 150 phase 1 through 4 clinical trials in over 48 countries. Our data is incredibly unique in that it is predictive, and this really enables an understanding of participants in your trial, unlike any other technology. We capture medication adherence via directly observed therapy that is reviewed by our proprietary AI, and we have demonstrated that this method of adherence capture is directly correlated with PK. Therefore, you can have extremely high confidence in the adherence data that's collected through our app. Additionally, we have found that participants' first 14 days of medication adherence data captured with AiCure is predictive of their overall medication adherence at the end of the study, and it's predictive of early termination. So that means you can identify which participants are likely to be non-adherent and which participants are likely to not complete the trial with just 14 days of data. The literature and our research also demonstrate that timely and effective intervention can modify participant behavior to either improve or restore adherence. And using this, we've developed a mechanism for effective central monitoring of AiCure adherence data and prompting appropriate operational action or intervention across all the stakeholders, so the site, the CRO, the sponsor, and AiCure, that actually leads to improvements or restoration in medication adherence rates, data collection outcomes, and even retention. Furthermore, through our digital biomarkers, this DBM data can predict future assessment scores and outcomes, and that frequent and longitudinal digital observations can provide a robust picture of patient health and behavior states. So these data can support analyses of treatment response, identifying patient subtypes in correlation to clinic assessments. And if our digital biomarkers are coupled with adherence monitoring, you know that your digital measures are taken at a time when the participant was adherent to the study drug, which is really essential to understanding digital biomarker results. So diving a little bit deeper into our biomarkers, our platform collects multimodal digital biomarkers, which mean the app simultaneously captures both audio and visual data. This is low burden and high yield, so that really gives you brief information-rich windows into patient behavior. And some examples of that are open-ended questions for collection of free speech and spontaneous facial expressivity, or responses to images for collection of behavior in response to stimuli. And we deploy bespoke projects that ultimately support development of validated, scalable tools. And here's how we do it. So our app records participant audio and video during a digital biomarker assessment, for example, a picture description task like you see here on the screen. And then our computational pipeline automatically extracts frame-wise digital measures and processes the natural language, and the result is a derived summary of multimodal digital measures, including vocal, facial, speech, and movement. So in collaboration with Karuna, which is now BMS, we deployed our digital biomarkers and adherence duo in a longitudinal phase 3 open label extension study of schizophrenic patients. And in schizophrenia, the positive and negative symptom scale, or PANSS, is gold standard for evaluating the efficacy of antipsychotic drugs in clinical trials, and consists of clinician interviewer probing the patient with leading questions and rating systems, both in verbal response and body language. And in this study, we deployed a digital picture description task in AiCure using positive, neutral, negative, neutral image series and a normative valence arousal rating. 10 of 14 time points of this digital biomarker assessment aligned with in-clinic PANSS scores during the treatment period. And in this study, over 16,000 descriptions were collected across 229 individuals with schizophrenia. And we completed 2 joint publications with this study. The first joint publication, we sought to examine the effect of picture emotional valence in digitally captured measures of speech, vocal acoustics, and head and facial movement in patients with schizophrenia using the digital picture description task. And we found that 6 of 11 of these digital measures were modulated by emotional valence. So positive picture responses had less upper facial movement and angular head movement. Negative picture responses had higher pitch and reduced pitch variability relative to the positive picture responses. And the results of this publication asserted that we can modulate multiple digital measures and that negative and positive stimuli can evoke valence-specific effects on audiovisual measures relative to neutral stimuli. And these findings really gave us the foundation for future work on the ability to digitally capture emotional modulation of facial expressions and head movement in schizophrenia, which is key to the PANSS assessment. So in the next joint publication, we used that information as a foundation to determine the predictive capability of these audio and visual digital assessments collected through AiCure to predict PANSS. In doing so, we use generalized estimated equations, or G, which leverage longitudinal data sets by modeling within subject correlations separately, resulting in parameter estimates that can indicate both the direction and the strength of relationships between digital measurement predictors based on available data points. And we leveraged over 1,000 longitudinal data points to determine the relationships of these measures for the PANSS factor score. And we found that the measurements were predictive of PANSS. So the negative symptoms were associated with reduced emotional facial movement, psychomotor slowing, and reductions in expressive vocal periodicity measures, where the positive symptoms were associated with increased emotional facial, head movement, and increased vocal periodicity. And so this really means that these brief remote assessments can predict subscale scores within the context of a clinical trial, which is extremely powerful in terms of patient burden, cost, and scalability. So in conclusion, digital biomarkers can support structured data collection in a low-burden, high-yield mechanism that enables frequent longitudinal and ecologically valid observations. And these data are indicative of patient health and behavioral state, and it's especially powerful in evaluating treatment response when collected with high-confidence adherence data. Novel machine learning AI development can address challenges around diagnostic assessment or treatment optimization and could even be adopted in the real world, all of which can help accelerate drug development and approval of new medicines. That's all. Thank you.
Thank you, and we have time for a question or two.
Do you have any insights on patient compliance on using the app and providing data in the real world, as opposed to in clinical trial settings where they may be more incentivized to use it?
Yeah, so we've deployed this within clinical trials, but also in commercial spaces as well. And the patients are really compatible with completing the digital biomarkers in the app. The technology itself is built on habit stacking principles, so there's basic reminders in the app, but it's also structured so that we can deploy multiple features all at once within the app to really help the patient build a routine. So we think that that's really attributable to high adherence or high compliance rates. But then in addition to that, we've more recently developed a central monitoring structure, which then supports on top of that, either led by our team or in partnership with a clinical team, for instance, in a commercial setting that can really check in on your patients remotely to ensure that compliance.
And is there a potential to use it as a predictor of adherence in patients that already are taking it in commercial place?
Sorry, can you say that again?
Is there a potential for it to be used as a proxy for adherence outside of clinical trial as well?
Yeah, definitely. So we actually are exploring that right now. We have some commercial uses even in tuberculosis with government-funded contracts and collecting adherence data that way. Yeah.
And then I see we also have a question from the online audience. Can patients see their own data, and have they expressed interest in this?
Yeah, that's a great question. So in a clinical trial, we typically have the patients come into the visit, and then they work with the site coordinator, who then surfaces the data and provides them feedback. That's part of our central monitoring structure, where we actually are encouraging sites to intervene with their participant. And intervention doesn't always have to be a negative intervention, meaning, hey, you forgot to take your pill 3 days this week. It can be a positive intervention as well, and really encourage the participant to keep going. And if you have 100% adherence, great job, here's your feedback.
Great, thank you so much, Megan and Ryan. All right, 2 more.